Hi everybody, it is your AP Biology teacher, Mr. Poser. We are back. I'm going to be doing a series of unit recaps here throughout the course of the year because I put out a little poll and I asked, hey, what kind of videos should I make? And this is what most of y'all decided is that we should be doing recaps. So here we are. This is Unit 1, Chemistry of Life recap. These are the 6 topics in AP Biology. Unit 1, 1.1, Structure in Water and Hydrogen Bonding. Number 1.2 is Elements of Life. 1.3, Introduction to Biological Macromolecules. 1.4, Properties of Biological Macromolecules. 1.5, Structure and Function of Biological Macromolecules. And number 1.6 is on nucleic acids. All right, so throughout this video, I'm going to be trying to be even more brief than I am in my regular topic videos, and just kind of breezing through this, basically going over the big things from each of these topics, the big ideas, and then moving on. I'm going to try and keep this video brief and not try to ramble that much. Okay, so here we go. Starting with 1.1, water is an amazing molecule. Why is it unique? Water is polar. And what makes water polar is that it has an uneven distribution of charge in one singular molecule. Okay, so here's a water molecule. It's made of 1 oxygen and 2 hydrogen atoms bonded together by covalent bonds. And water has a high electronegativity, which means that it kind of gives it a slightly negative, partial negative charge on one side of the molecule, and hydrogen is not, and that makes it slightly positive on the other side. Okay, so this is a very, very simple molecule, just H2O, and it's pretty easily naturally occurring, but it's polar, okay, because of its unique shape and unique electronegativity and difference in charge. Okay, and because water is polar, it allows it to do some stuff that nonpolar molecules can't. So what is a nonpolar molecule? Like this one, like oxygen gas, where there's an even distribution of charge. Okay, oxygen is slightly negative, so it's this other oxygen, okay? And there's an even distribution of charge in that molecule, hence why it is called nonpolar. Okay, so there's a whole group of biomolecules that are pretty much all nonpolar, and they don't mix well with water. In other words, they are hydrophobic. Hydro meaning water, phobic meaning fearing. So yeah. An uneven distribution of charge of a single molecule is what makes it polar, and because it is polar, it forms hydrogen bonds, and this is where water gets interesting. Hydrogen bonds aren't really bonds, so to speak, not like covalent bonds or ionic bonds. They're intermolecular forces, and there's kind of weak attractions between molecules. But hydrogen bonds, one hydrogen bond is not very strong by itself, okay? But when you have billions of water molecules in one single drop of water, okay, there are billions and billions of hydrogen bonds forming and breaking every second. And when you have a lot of those bonds together, that's what makes water pretty strong. It makes it pretty sticky, in other words. And these hydrogen bonds are really going to keep these water molecules together. And this picture is showing you here that, look, here's an oxygen that's got a negative, partial negative charge, and it's attracting, okay, because opposites attract, right? Negative and positive attract each other. And it's attracting these positive hydrogens over here, okay, and this hydrogen's on the same water molecule is attracting the negative oxygens over here, okay. So these hydrogen bonds are what gives water its super unique properties, okay, and allows it to do a whole bunch of goofy stuff in comparison to other molecules, like this, okay. A few of water's properties is that it has a high evaporation temperature, a high boiling point. It's sticky, okay, that has cohesion and surface tension, and it has adhesion. And all 3 of these properties can be explained by hydrogen bonding. So high evaporation temperature means that it takes a lot of energy in order to turn liquid water into gas, okay, or into water vapor. All right, when you heat something up to its boiling point, a fluid up to its boiling point, you are separating the molecules enough from one another, okay, that they start to, fly around, and they have a whole lot of, because that's what temperature is, the kinetic energy of particles. They start to fly around and take up a whole lot more space. Okay, it's still a fluid, but the molecules are very spread out from one another. Okay, and because of those hydrogen bonds pulling on the water molecules all the time, it takes a lot of energy in order to separate them and break those hydrogen bonds. So water has a high boiling point. Water is very sticky. Cohesion and surface tension, those kind of tie together. Water molecules tend to stick together a lot, and it requires a lot of force to break those apart from each other. And if we're talking about the surface of water, okay, certain animals are able to, kind of glide on the surface of water, like lizards and bugs and that kind of stuff, glide on the surface of water because it has a very high surface tension. And that's because of those hydrogen bonds keeping those water molecules kind of close together, intact. And water, a lot of hydrogen bonds makes it very strong. And then, of course, adhesion means that water is going to stick to other polar surfaces very well as well. It's just water is very sticky. That's how my biology teacher described it. Okay? And living things rely on these three properties, that it's not going to boil easily, it's not going to break apart easily, and it's going to stick to polar surfaces. Okay? For example, plants would not be able to use capillary action to drive water up from their roots to the top of a very tall tree without water being very sticky and without water sticking together. Okay? All right, moving on to 1.2. Living things are made of carbon. Okay? The elements of life, the primary one is carbon. And why is carbon so special? We are carbon-based life because carbon has 4 valence electrons. It's a pretty small atom, and it has 4 valence electrons, which allows these atoms to form 4 different bonds and start to make these really, really big complex molecules that living things rely on. It makes it a really, really good building block. So a lot of the molecules that we're going to be looking at in this unit have carbon at the center of it, literally at the center of it. And that's a really big deal. Okay, that's why carbon is really good for making biological molecules. Okay, perhaps carbon is so important that we even categorize chemistry and molecules into 2 different groups on whether or not it has carbon. Organic molecules are found in living things and contain carbon. Inorganic molecules are not typically found in living things and do not contain carbon. So these molecules that we're going to be looking up here in a second, there's all organic molecules, but something like salt or something like even oxygen or maybe like hydrogen gas that doesn't have carbon, it's needed by living things, but it's not found in living things. It doesn't make up the cells of living things, hence it is inorganic. So like if you grow organic bananas, that means you don't use any inorganic compounds in order to grow that banana. Okay, that's what it means by organic food, is that it's all molecules that come from living things to help it grow. All right, couple more things in 1.2, the composition of biological macromolecules. Here's our 4 main titans here. Carbohydrates, lipids, proteins, and nucleic acids, and these are the elements that comprise each. Carbohydrates, carbon, hydrogen, and oxygen. Okay, you can see only those 3 atoms in a whole bunch of these monosaccharides over here. Those are all very, very simple sugars. Lipids have carbon, hydrogen, oxygen, and phosphorus in phospholipids, which is a very important type of lipid that we're going to talk a lot about in Unit 2. And proteins have carbon, hydrogen, oxygen, nitrogen, as you can see from this amine group over here in this amino acid, and sulfur. Some amino acids have sulfur. And then nucleic acids have carbon, hydrogen, oxygen, nitrogen, and phosphorus. Okay, so here's our 4 main biological molecules. Those are the atoms they comprise of. Last part of 1.2 is about isomers, and isomers are molecules with the same numbers and same types of atoms, but with different structures. And there's a few different types of isomers that need to be known for the AP exam. Okay, the first of which is called structural isomers. All right, maybe you can find it yourself, but these 2 glucose molecules, they are both glucose. They are both C6H12O6. They all have the same atoms, the same composition, but they are 2 different molecules. They are isomers of one another because of the arrangement of the atoms. There's slight difference in the structure between these 2. Hence, these are what we call structural isomers. Another type of isomer that is going to be important is what we call a geometric isomer, or what's also called a cis-trans isomer. And those are isomers that have the same, chemical formula, same atom, same number of atoms, same types, and they're the difference of their arrangement around a double bond is different, hence why they're called geometric, right? So this is what we would call a cis, or it means the same side of a double bond, and this is what we call a trans isomer, where two groups are on opposite sides of a double bond. Okay, so you can see the HC3 is on the opposite side of these other ones over here, okay, but on this one they're on the same side. Hence, those are geometric isomers, cis and trans. And then the last one is super interesting. They're called enantiomers, and that's what we call mirror image molecules. Okay? And they're very similar to structural isomers, but these are special because they are mirror images of one another. Okay, here's D-glucose and L-glucose. By the way, this is another example of a structural isomer because glucose can also be linear or cyclic, like this. Okay, it's a cyclic in a cycle, or it's in a line like this. All right, and these are enantiomers. Okay, these look exactly the same, but they are not. If you put them on the same side as one another, okay, they have a different arrangement of atoms. Okay, so a lot of times enantiomers are named with a little letter that indicates their direction in front of it. But yeah, that's 1.2. Let's move on to 1.3. There's 2 main things you need to know about 1.3. Well, I guess 3. is that macromolecules are very large molecules. Macro is the opposite of micro. Macro means big, and they're composed of many atoms. And these atoms comprise what we call monomers, and monomers are molecules that are smaller subunits of macromolecules. Mono means one. Okay, so if I take a monomer and combine it, a whole lot of them, into a really big molecule, okay, I am making what's called a polymer. Polymer means poly means many. Okay, so these big molecules that we're going to talk about, like a nucleic acid, like DNA, or like a protein, or like a polysaccharide, or like a big old lipid. Okay, those are macromolecules. Those are polymers of these smaller molecules called monomers. So like this one right here, this is an amino acid. It is the monomer of a protein. If I take a whole lot of amino acids and link them together into a chain, then I'm going to start to build a protein. And proteins can be described as many of amino acids linked together, a chain of amino acids, so to speak. So an amino acid is a monomer, and a protein is a polymer. Okay, and in order to make a polymer, okay, it requires a type of reaction called dehydration synthesis, linking two monomers together. And how this happens is that one hydroxyl group, or an OH, is removed from one molecule, and one hydryl, or just an H, is removed from the other, creating a new covalent bond and a new H2O molecule. Okay, so let's just say I have let's say start of a polypeptide over here. There's 3 amino acids there, and there's an OH on one side. And then here's another amino acid. I'm trying to link this amino acid to these other 3, and this has a hydryl group. Okay, they can use an enzyme in order to help me make a water molecule. Okay, using that H and that OH, because H2O, right? And then, okay, since I remove these two, these two H and OH, all right, I'm forming a new bond between these amino acids, and there you go. I'm linking them together. Okay, so this is the step of linking together monomers in order to form a polymer. This is what we call an anabolic reaction, but it is what we call dehydration synthesis. Dehydration is like, oh, I'm thirsty. Yeah, but like we're taking away a water molecule. That's why it's called dehydration. And then the opposite of dehydration synthesis is what we call hydrolysis, or breaking a dimer, or which is 2 monomers, or a polymer into monomers requires the opposite of dehydration synthesis, where we're taking a water molecule and it quote-unquote attacks a covalent bond, leaving an OH on one molecule and an H on the other, and it's the exact opposite. So like, there's no other way to say it. It's the same thing except reverse. Okay, so this water molecule comes in and kind of destroys this bond between the 2 molecules, drops off an OH on one molecule and an H on the other, and there you go. That covalent bond is separated, and now you're breaking down a big molecule into smaller ones. So when might you need this? Well, if you have a polysaccharide that's stored carbohydrates in your muscles, okay, and then all of a sudden you need more glucose, okay, or it's in your liver as well, and you need more glucose in your blood for whatever reason, okay, your body can hydrolyze those polysaccharides and get the glucose out, so you can have some quick energy to use. Okay, let's move on to 1.4. 1.4 is all about the properties of biological macromolecules, the monomers more specifically. All macromolecules are made of monomer subunits. Okay, so each one of our 4 molecules, carbohydrates, proteins, lipids, and nucleic acids, are made of smaller molecules called monomers, okay, and we're going to learn what each of those monomers are and the properties of them. So the monomer of a carbohydrate is what we call a monosaccharide, and we saw this picture before. A monosaccharide is 3 to 6 carbons long, used primarily for quick energy through the process of cellular respiration, which we're going to get into in Unit 3, along with disaccharides. Okay, so monosaccharides, glucose being the most important one, fructose, maltose, deoxyribose are all examples of what we call monosaccharides, and galactose over here. Well, something like sucrose or lactose, those are what we call disaccharides. Di meaning 2. You link 2 rings of carbon together. And sometimes these can be linear as well, as we looked at before too. Okay, but either way, these are called monosaccharides, and they're composed of C, H, and O. The monomer of a protein is what we call an amino acid. We saw this picture already too. And so if I take a whole boatload of amino acids and link them together into a chain, I start to make a protein. Here's the thing about amino acids. All amino acids have what we call an NH2, or an amine group, and a COOH, or a carboxyl group, and a central carbon. So you look at this amino acid. There's an NH2 right there. That's called an amine group. There's what we call a central carbon, and then we have a COOH on the other one. So when you're looking at a picture of an amino acid, it's a giveaway to see the NH2 and the COOH. This is an amino acid every single time. Okay, and these three components here, the COOH, the NH2, and the central carbon, or what we call the backbone of an amino acid, and that's going to become important here in a bit. All amino acids have that backbone, but there's 20 different kinds of amino acids, and the way that those amino acids are different is what we call their R group or their side chain that might have different properties than each other, right? So there's some amino acids that have a nonpolar side chain. There's some that have a polar side chain. There's some that have a positively charged side chain, and then there's others that have a negatively charged side chain. And the interactions between the side chains of those amino acids is going to come back in a big way here in 1.5. But that's going to be important. All right, so you have the backbone, the amine group, the central carbon, and the carboxyl group, and then you have the side chain of one single amino acid. All right, and a protein could be like thousands of amino acids long. All right, so when you put those two together, this goofy stuff starts to happen. We're going to see that in a sec. Okay, let's talk about lipids. This important note about lipids that all lipids are nonpolar. They're hydrophobic. It is a nonpolar molecule. Okay, and the monomer of a lipid is a fatty acid. And you may have heard of fatty acids before. There are saturated fatty acids and there are unsaturated fatty acids. And there's pictures of them over here. So all fatty acids have a carboxyl group, kind of like an amino acid, and a carbon chain. These squiggles here are just carbons and hydrogens. Yeah, it's, organic chemistry. You use so much carbon that you just kind of use a squiggle as a shorthand to replace it. So these are all carbons and hydrogens over here. All right, and what you can see is that saturated fatty acids, this one's saturated, all has single bonds in its carbon and hydrogen chain over here. Okay, those are all single bonds, but an unsaturated fat has at least one double bond. So here's what we call a monounsaturated because there's one double bond in that carbon chain, and here's a polyunsaturated fatty acid because there's multiple double bonds in that carbon chain. All right, and the fact that saturated ones do not have double bonds, unsaturated fats do have a double bonds, lends to their properties. Okay, and I think I talked about the properties here. Oh, I talked about the properties in the next one. Okay, another two types of lipids that are going to be important: triglycerides. Those are fat molecules. Okay, so like the way that your body stores lipids in your blood or transports lipids in your blood is called a triglyceride. Those are composed of 3 fatty acids and one what we call glycerol molecule. Hence triglyceride. Okay, tri meaning 3 fatty acids. While phospholipids, which we're going to come back to in Unit 2 big time, which make up cell membranes, have 2 fatty acid tails, 1 glycerol and 1 phosphate group. So here's your fatty acids, here's your glycerol, and here's your phosphate. Here's a triglyceride or a fat molecule, here's the glycerol, and here are 3 unsaturated fatty acids, saturated fatty acids, although they can be unsaturated as well. Okay, so then moving on to the last monomer, it's called a nucleotide. So a DNA molecule is made up of long chains of what we call nucleotides, and that's the monomer of a nucleic acid. Nucleic acids are made of 1, if it's RNA, or 2 chains of nucleotides, and each nucleotide all has the same 3 things. They have a 5-carbon ring that could be deoxyribose or ribose, so it's basically a carbohydrate, right? It's basically a monosaccharide. It has a phosphate group, and it has what's called a nitrogenous base. So you can see all those nitrogens and carbons interacting in those 2 rings with each other, and that's gonna give it some properties here that we'll talk about in 1.6. Okay, but all nucleotides have these 3 components, and that's going to come back later. There's 2 types of nitrogenous base. Pyrimidines, like cytosine up here, have 2 rings of nitrogen, so like the one that we saw up here. Oh, sorry. Pyrimidines have 1. Pyrimidines have 1, and purines have 2. So hang on a sec. I swapped these. There we go. That's cytosine. That's adenine. Adenine is a purine. Purines have 2 nitrogen rings. Pyrimidines have 1 nitrogen ring. So this is cytosine. I swapped those labels. My bad. But pyrimidines are cytosine, thymine, and uracil. Purines are adenine and guanine. So adenine and guanine have 2 nitrogen rings, and pyrimidines only have 1. Okay, let's move on to 1.5. We're almost done here, team. Complex polysaccharides. So this is the polymer of a carbohydrate. All right, we're talking about monosaccharides before. When you link them into a chain, that's called polysaccharides. They're made through dehydration synthesis of monosaccharides. And what polysaccharides are typically used for, they're used for energy storage, like say, for example, plants store energy or carbohydrates in the form of starch. Animals store it in the form of glycogen. but polysaccharides can also be used for structure. So plant cell walls, which we're going to look at next unit, are made of cellulose, and chitin is an example of a polysaccharide that makes up the exoskeleton of insects. So there you go. When you step on them and they go crunch, that's why, because they're polysaccharide exoskeleton. Okay, so there you go. A polysaccharide, if you can hydrolyze it into monosaccharides. All right, so that's what you need to know about polysaccharides. All right, proteins though. Proteins, this is where it gets a little complicated. Okay, as I said a bunch of times already, proteins are long chains of amino acids, or what we might call polypeptides, attached by peptide bonds. And what a protein does, proteins do pretty much everything. They have so many different functions. There's so many different kinds of proteins. And the reason why is that proteins are so varied because of the way they're shaped and the way that they fold over one another. These amino acid chains fold over one another. And this structure of a protein can be studied at 4 different levels here that determine how it's shaped and how it folds, and thus, thereby, what its function is in living things. And we're going to look at so many types of proteins in this class. Okay, so if we're studying the primary structure of a protein, we are simply looking at a protein sequence of amino acids. All right, so if you see a picture of a protein where it looks like this chain of amino acids, you are studying its primary structure. All right, so you can see this phenylalanine, leucine, serine, cysteine. That's studying its primary structure. Okay, just a sequence of amino acids. Now, if you're studying the secondary structure of a protein, once you get that chain long enough, it starts to fold or coil into what we call an alpha helix or a beta pleated sheet. Okay, so here's your primary structure. Here's a chain of amino acids. It starts to form these goofy shapes. And the reason why it's forming these goofy shapes, these helices and these kind of zigzags, is because of the hydrogen bonds, hydrogen bonds, and the amino acid backbones. Okay, so we looked at amino acids before. There was a central carbon, an amine group, the NH2 and the COOH. Guess what? Those are polar, right? And they form hydrogen bonds. Okay, and because of the hydrogen bonds, they start to form these goofy shapes in secondary structure. Okay, and it starts to get even more goofy once you get to tertiary structure, when the pleated sheets and the helices start to kind of fold over one another and form this big blob. Okay, then you're studying what's called tertiary structure, and it starts to form this big blob due to what we call side chain interactions. So that other part of that amino acid, that R group. Okay, remember how I said they can be polar, they can be non-polar, charged, or positive or negative, right? Okay, this is what lends itself to tertiary structure, right? So if I'm folding over this polypeptide, I got a polar amino acid on one side of a fold and a polar one on the other side. Well, polar molecules like each other. They tend to stick together, hydrogen bonds, right? So they're going to start to attract to each other. Or if it's in polar and non-polar, they might like pull away and form a different shape. If something's positive and negative, they're going to attract. But if it's positive and positive across from each other, then they're going to push away from each other. Okay? But the main thing that you need to know is that the way that the helices and the sheets kind of fold together and start to combine is because of side chain interactions. Okay, and that's the main thing about tertiary structure, right? Is that you're starting to get a functional polypeptide there, because it's starting to fold and form this unique shape because of those R group interactions. Okay, speaking of tertiary structure, all right, if you mess with the tertiary structure of a protein too much by changing the temperature or the pH, it starts to mess with those side chain interactions and change the properties of amino acids, and it's going to cause a protein to basically unfold and break, and that's called denaturation. That'll come back later this year as well. Okay, so if you denature a protein, it will not work, and you're basically disrupting those tertiary structure, those side chain interactions. All right, and quaternary structure, this is the most complex form of protein structure. All right, quaternary structure of a protein is where you have combinations of multiple polypeptide subunits or multiple polypeptide chains. So for example, the main one that I go to is hemoglobin. Hemoglobin is a protein that carries oxygen in your blood. It is made of 4 different polypeptides put together into one big protein. Okay, so that is studying what we call the quaternary structure of a protein. All right, last piece, 1.6. We got to talk about DNA a little bit and RNA and the differences between them. DNA, all right, my friend right here, it's broken a little bit, but DNA is made of 2 polynucleotide strands. All right, so we have 2 long chains of nucleotides that twist together and form what we call a double helix. Okay, but not what's interesting about the double helix is that, well, these 2 strands are what we call anti-parallel, and the nucleotide chains that are oriented in opposite directions of each other, right? So like I said, you have two long chains of nucleotides, but they're like and they form kind of a ladder shape. Okay, except the ladder shape, one side of the ladder is kind of upside down in comparison to the other one, which is interesting, right? It's anti-parallel, and the word that we use to describe its the directionality of these nucleic acids is that you have a 5' end on one side and a 3' end on the other, and those numbers are named after the numbers of carbons in a deoxyribose sugar in the middle. All right, we're going to take a look at that in a sec. Okay, but you have a 5' to 3' end. All right, running one direction, and then you have a 3' to 5' in the other direction. So these two molecules are kind of like, they're anti-parallel to each other. They're parallel, they're never going to intersect really, but they're running in opposite ways. They're upside down to each other. Okay, that's what makes DNA anti-parallel. So another thing to know about DNA is that the nitrogenous bases, okay, we looked at a nucleotide before. Every nucleotide has a deoxyribose sugar, a phosphate, and a nitrogenous base. And the nitrogenous bases are perpendicular, okay? They form a 90-degree angle to the sugar-phosphate backbone. So I drew a kind of a red line here showing that these bases that are kind of meeting in the middle, okay, as you can see from my model here, like T is meeting up with A, C is meeting up with G, okay? They are perpendicular, form a 90-degree angle with the backbones, the sugars and phosphates of the nucleotides in a nucleic acid. Okay? Couple more things to know about nucleotides and the structure of nucleic acids in comparison to one another. The 5' and 3' end of DNA molecules are named after the numbers of carbons in the pentose ring of a nucleotide. Okay, so I said I was going to come back to this. Here it is. So here's the phosphate group of a nucleotide. Here's another phosphate group of another nucleotide. Okay, like I said, big long chain. Here's what we call the 5' carbon. Here's the 3' carbon. Okay, and we can number the carbons 5, 4, 3, 2, 1. In this way, and that's what gives us our directional terms for these two chains of nucleotides. Okay, so here's your 5', here's your 3'. The phosphodiester bond, that's the name of the bond between between nucleotides to form a nucleic acid. All right, so if I use dehydration synthesis between nucleotides, hey, I can link them together to form a nucleic acid. I'm forming phosphodiester bonds. But anyway, RNA, okay, the difference between RNA and DNA. First one is that RNA has a ribose as its pentose sugar, pentose meaning 5 carbons, while DNA has deoxyribose as its pentose sugar. Okay, so RNA has an OH or a hydroxyl group on the 2' carbon. DNA doesn't, hence deoxy. All right, so I can tell just by looking at this that this is a DNA nucleotide because I'm looking at this 2' carbon over here, and it does not have an extra oxygen. It does not have an extra what I call hydroxyl group right onto it. But this one would be RNA, okay? So that is one key structural difference between the two. Another one is that RNA contains uracil rather than thymine, okay? So the 4 letters, the 4 nucleotides that you can find in DNA are A, G, C, and T, but in RNA it is A, C, G, and U, okay? So the base pairing rules state that A always matches up with T, and C always matches up with G, or as I like to tell my students, the apple goes in the tree, the car goes in the garage. Okay, but when it comes to RNA, A matches with U. Okay, apple goes underwater or something like that. When you're matching these up, but that's going to come back a lot more in Unit 6, especially in topics 6.1 and 6.2 and 3. Okay. But yeah, that's another key difference between them. And then the last key difference between RNA and DNA is that RNA has one polynucleotide strand. It is made of one chain of nucleotides, while DNA is 2 that are coiled and twisted and bonded together thanks to hydrogen bonds in the middle between these nitrogenous bases. Okay? And like I said, the base pairing rules apply in DNA. A always matches up with T, C always matches up with G. And I believe that is it. Yeah, that is it for Unit 1 recap. I tried to go through everything as fast as I could while still trying to hit the main points. If you have any questions or if I missed anything important, please let me know in the comments, whatever. I'm going to be doing more of these throughout the year, and I hope this is really helpful. Have a great rest of your day. We'll see you next time.